Okay, thank you. Welcome everybody at this meeting. It's a pleasure to introduce Katerina Sam as our speaker and historically first online speaker at GTO plenary. She will be talking about the interaction webs in forests, and that's part of the large research program she is leading, stretching from Australia all the way to Japan, as you will see. Katerina started studies in this direction during her PhD studies, which took place mostly in Papua New Guinea. And that was when she was primarily concentrating on ornithology research and biodiversity of birds, and secondarily what they are doing in the rainforest ecosystems, especially what they are doing to insects. This is a step further, I would say, in the right direction from her master's studies, which were completely on birds and behavioral ecology. But since she then transferred to the Institute of Entomology of the Czech Academy of Sciences, where she's leading her laboratory now, incidentally at my department of ecology, then I think that was a good stimulus to focus not only on birds only, but especially on birds and what they are doing to insects. And today we will hear, I suppose, about all these complicated interactions, where of course also plants are playing their role, as we can see from the title. So very pleased to have you, Katerina, as our guest.
Thank you very much. It's really my honor to be here. Despite I'm in my office, I have the ability to talk to so many people online, somewhere on the other side of the screen. And today I am going to talk about the topic which I really love and which I really focus on over the last several years. And it will be about the predators, their predation rate, about whether and where they are letting the plants to flourish, or whether the plants by themselves are super disgusting, protecting their own foliage. And I don't know whether you can relate to my experience, but whenever I feed my kids some green stuff on the plate, they usually spit it in disgust. And whenever I was doing some experiments, feeding experiment with caterpillars, I was observing the very similar things. The caterpillars didn't like it. Sometimes they ate it very well, but sometimes not. And I was wondering whether the plants by themselves are able of their own protection. And this aspect is called the bottom-up control. Generally, the thing that plants by themselves, by production of huge amount of biomass and by protecting, by having their own defenses, are controlling the higher trophic levels. The other aspects and perspectives in the ecology are saying that the predators and the higher trophic levels, like herbivores, are determining the biomass of the lower trophic levels. And this idea was first time published in 1960 in the short paper in the American Naturalist. And from the time it caused a lot of discussion and brought the support for both top-down and bottom-up control aspects of the ecological theories. And we can see today that the truth is somewhere in the middle. We have seen the support for the bottom-up control, for the top-down control, and I have to say that the bottom-up control is usually determining if there is going to be some green foliage at all on the study site. And the top-down control is generally shaping the shades and the type of the vegetation growing at the site. And during the evolutionary race of the plants and the insect, the plants developed the two types of defenses, which we can split as constitutive and induced. The constitutive defenses are always present in the plant, and that's why they are useful when the attacks are repeating often and where this is a big abundance of herbivores. Induced defenses, on the other side, are induced only after the herbivore's attack happens to the plant, and that's why they are usually cheaper and useful when the attacks are not predictable in the time. And finally, the induced defenses might be split into those direct and indirect. The direct defenses are aimed against the insect directly, making them very sick or even dead. And the indirect defenses usually work through the compounds, chemical compounds, which are released to the air, which are attracting the natural enemies of the herbivores and providing thus the reward for the predators and helping the plants to flourish. And you might think that there is a trade-off between such defenses, between the direct and indirect, because if you look at the tree, which has very strong direct defenses, there will be only few very sick caterpillars feeding on them. And when we look at the tree with the very weak direct defenses, there will be maybe many very happy caterpillars feeding on that tree. And if we are looking one trophic level higher, such a tree with very strong direct defenses will be producing very short and small signal to the natural predators. And the tree with abundances of the insect will be producing a very strong signal. It's also very useful for the birds to arrive to the tree, which is providing a bigger reward in the food. And we were therefore wondering how the birds actually find the herbivore-rich and potentially herbivore-rich trees. And because we all know that the predators orient themselves visually, we were testing in the tropical forest where the understories are usually very dense. If the plants can orient themselves according to the visual image on the leaves. So this is what we were doing along the elevation gradient of Mount Wilhelm in Papua New Guinea, and we were tearing apart pieces of the leaves, producing the saplings which had herbivorous damage and did not have herbivorous damage. And we saw at least some of the study sites, the birds were more attracted and were searching on the trees which had higher herbivorous damage visually visible on them. But because the effect was not very strong, we were wondering what are the other aspects attracting the predators to the herbivore-rich trees. And here are the results of experiment which my PhD student Anya Mrazová did. She was actually mimicking the high herbivory by application of methyl jasmonate, which is a hormone which is regulating the production of herbivore-induced chemical compounds, which are very similar to those which plants are producing after the plant is attacked by herbivores. So after she sprayed the trees with methyl jasmonate, she saw in the temperate regions that the birds were attracted to the sessile as well as to pedunculate oaks. And the same method worked really well also in tropical understories with the forest, with the Ficus haliana along the elevational gradient. And we were further wondering whether the birds have this ability to orient themselves to herbivore-rich trees innate or whether they need to learn it. If they need to learn it, how they do it. So we conducted this experiment in Czech Republic again with the gratids, temperate birds. We took very naive babies from the nests. They had no experience with the foliage yet, and we split them into 3 groups. One group of the gratids was taught to associate the smell of the herbivore-induced tropical guavas. And the second group was learned to associate the food reward with the smell of herbivore-induced elms. And finally, the third group was left without the training. They were totally naive to the leaf green foliage. And we were assuming that if the birds are innate, they will not react, they have the ability innate, they will react to any type of the herbivore-induced trees. If they are capable of generalization, they will prefer any herbivore-induced tree if they are trained to some of them, but the control birds will behave randomly in the experiment. And finally, if the birds are learning one odor at a time, they will be only following the smell of the tree they were trained to. And this is what we saw in the control babies. First, we were offering them random variations of the induced elms, guavas, induced guavas. And in the experiment, they were behaving very randomly, flying from one tree to another, spending 50% there and 50% on another sapling. But when we were looking to the birds which were trained to something, we saw that they are significantly attracted to the plant, herbivore-induced plant to which they were trained to. But when we offer them other plant species, they didn't prefer the type of the tree without the herbivore's induction. This all means that the birds do not have the ability to smell the herbivore-induced trees innate. They have to learn it, and they are specifically learning one odor at a time, which makes them very flexible, ready to react to the pest outbreaks whenever they appear. As soon as the birds are associating the food reward with the smell, they can learn it very quickly and react. So the birds react visually to the visual cues left behind by the herbivores. They react to the chemical cues left by the herbivores, and they learn them flexibly. They are also omnipresent, and they consume the huge amounts of arthropods yearly. But still, are they really good predators? Because many people argue that their biomass globally is very small. In contrast to the arthropods, predatory arthropods like ants and spiders, who have much bigger biomass globally and which are estimated to consume even more arthropods and tons of the insect annually than the birds. So we were thinking about how to study the predation. And I have to say this is usually very difficult because we are dealing with the action which is happening somewhere in the dense foliage, usually somewhere above your head, and to study the predation is generally very difficult. That's why some non-direct methods were used to survey or estimate the predation rate. People were pinning some dead or larvae on the leaves, measuring how fast they disappear. There were the other methods where people were pinning the plasticine caterpillars. The disadvantage and advantage of these methods is that when the real prey is pinned somewhere and disappears, we can't say what kind of predator ate it. With the plasticine, it's a little bit better because the bite marks usually stay in the print in the plasticine, so we can say whether the arthropod or bird was predating. But both these methods are either underestimating or simply not measuring well the willingness and the real ability of the predators to remove the live insect from the system. Because the prey is not fighting back, doesn't provide the correct chemical cues, and simply is not behaving the way as normal prey does. I believe that the better way how to measure the predation is by the exclosure of the predators from the system. In this way, you are removing the predator, and the insects which would be normally eaten will be surviving on the foliage. And at the end of the experiment, you can measure what survived and what would be then normally eaten by the predator. And as I was digging into how to study the predation correctly, my naturally occurring questions were where the trophic cascades appear and where do the predators express the strongest effect on the lower trophic levels. So the traditional view says that the terrestrial trophic cascades are strongest in tropics and at low elevation, generally at the places where the productivity is the highest. But there have been several reviews which show that the cascades might be strong in the moist wet regions, in the cool regions, generally making kind of a U shape along the latitude, or in the temperate regions only. I was lucky to participate in the global study which used Plasticine caterpillars globally. And based on these results, you can see that the Plasticine caterpillars received many attacks in the tropics, in the lowlands, and the brown dots show that most of the attacks were caused by the arthropods. This would imply that really ants, wasps, the predatory arthropods are the most important predators globally, killing many of our caterpillars, and that the trend in the predation decreases towards the lower, cooler, a little bit rash conditions. In this study, we didn't find any effect, any pattern in the predation by birds, but there was many aspects of this study which needs to be mentioned further. And I was wondering whether I could get the same results if I look into the exclosure experiments. So as the number of predator exclosure experiments started to appear around 20 years ago, I decided at the time maybe to review them again and check whether the patterns observed in the Plasticine experiments can be confirmed from the exclosure experiments. So you can see that authors of the studies usually excluded ants, vertebrates, or vertebrates and ants together. They used various methods to do it. And I would like to mention also that many authors actually claimed and wrote that they are excluding the birds, but what they did is that they placed the cages around the foliage permanently, excluding actually several types of the vertebrate predators at the same time. So in total, I collected 475 experiments from 150 publications. I split them into those which actually remove the vertebrates and ants, and this is what we saw. So at the global scale from the meta-analysis, it looks like then when we exclude the predators of vertebrate predators, it has significant and strong effect on the arthropod abundances. The arthropod abundances increased about 3 to 2 times, but when the ants were excluded, the abundances of the arthropods didn't change significantly. But when we look into the feeding guilds, predatory arthropods and herbivore arthropods, in the exclosure of vertebrates, it looks like that the vertebrates work like the interguild predators, feeding on both types of the arthropods in this equal proportion. On the other hand, the ants seem to be having effect only on the herbivore arthropods, but not on the predatory arthropods. It might be the real thing that there is a competition between ants and mostly spiders. They're fighting, but the ants are not strong enough to remove them effectively from the system. Or it can be simply an artifact of the method, because by the methods which you are removing the ants, you are actually removing also the predators. Along the latitudinal and elevational gradient, we didn't find any strong pattern, but when we look a little bit down in the graph to the plant image, we saw that the exclosure of the vertebrates actually resulted to a rather weak cascading effect and didn't lead to the big change in the plant image. But the exclosure of the ants led to the high increase of the plant image. This is correlating well with the observations that ants are very likely affecting the herbivores. Herbivores are surviving when the ants are gone, and then the plant image increases about 2 times. And we observed the pattern to have a very steep increase along the latitude and along the elevation, with the effect being visible mostly in the lowest latitude and the lowest elevations. And because there was many problems in this meta-analysis, and we saw that the experiments which are so far available are not very comparable. There is an effect of the length of the experiment on the results, and the mesh size affected the results. We decided to work by ourselves, try to make the standardized method along the large scale, and we worked in Papua New Guinea along the elevational gradient of Mount Wilhelm, which is the highest mountain of the country. So this is how the mountain looks like. Here we finished the work at the tree line. I am standing in the middle of the gradient at 2,200 meters above sea level, and there is another 15 kilometers of the road to go to the bottom, to the coastline, to the lowest elevation. And along the elevational gradient, the habitat changed significantly from the very dense and dark, very high canopy forest to the very short forest with the open canopy and shrubs around 10 meters high, only while the canopy in the lowlands were about 30 meters tall. Also, the temperature decreased significantly along the elevational gradient from 25 to 7 degrees in average. And that's why we expected the predator communities to change significantly along the elevational gradient. And here we are getting back to the picture which you partly saw before. As I did the plasticine experiment there, you can see that the ants attacked majority of the plasticine in the lowlands. The predation rate decreased with the increasing elevation very steeply, generally correlating with the abundance of the ants, and the predation of the birds made slight hump-shaped or generally no change up to the mid-elevation, up to the 1,700 meters above sea level, and then the predation by them decreased very steeply. And knowing this, we decided to do the 6 treatments along the elevational gradient. We have got the saplings of the ficuses, which were not treated at all. The predators had the free access to them, and this was control trees with this C sign. Then we had the saplings from which we excluded ants by the tangle food. Then we had the saplings from which we excluded the predators by permanently standing cages around them, and combinations of these 2 treatments, tangle glue on the trunk and cage all around to prevent the access of all predators. And finally, those 2 tricky treatments to exclusion of the birds and bats separately. This is done by the cages which are covering the foliage crown of the sapling and which are open every day in the morning and in the afternoon to prevent and allow the access of the focal predators which needs to be in or out. So this is really demanding work because we needed to be there every day and attend all the cages every day. And this is how it looked like a little bit in the field, tangle glue on the trunks, cages around the foliage, sometimes hanging on the pulley system to allow us to pull them up and down. And the experiment actually used around 9 kilometers of the water pipes, nearly 1 full football field of the fishing nets. And here are the assistants building the exclosures in the forest understories. The experiment lasts 2 times 6 months. And after the 6 months and 12 months, we resurvey the ant abundances on the saplings by lowering the crowns of the saplings above the mosquito net, spraying that quickly by the mortain, slowing down insects, catching all of them by all the insects bigger than 1 millimeter. Here, assistants working, and you can see also the leaves being collected. And this was a real hell for me. The opening and closing of the birds and bats was tricky, but the herbivore is a very difficult task to do because the herbivore is generally always present in the tropical forest. And if we need to measure it correctly, we need to number all the leaves individually, measure the herbivore at the beginning, during the experiment, at the end of the experiment. That's why we numbered 30 leaves at each sapling. Then we photographed them, rescanned them, analyzed in Photoshop, analyzed in ImageJ, and got the herbivore change during the experiment. We, of course, needed to know a lot about the predator communities. We surveyed the ants, birds by myself, and we are still working on the metabarcoding of the prey which they are eating. And our students were helping us also with the bat survey. And when we look to the results, we can see a few interesting things, but also a big variance in our experiment. So first, when we look at the control, red lines, and the blue lines and exclosure, they are usually very close to each other. There is no big change. So ants altogether did not have any effect on the abundances of the arthropods. What we see in the green is the effect of vertebrate predators. When we excluded the birds and bats together, the abundances of the arthropods increased significantly in many cases, following kind of a ham-shaped pattern, which is correlating with the results which we saw on the plasticine caterpillars earlier. In the summary graph here, you can see that the controls do not differ from ants, and then the predators, vertebrate predators, and all predators are having a significant effect on the arthropod abundances. I promised you the results from birds and bat exclosures, but due to the logistical problems and difficulties in the field, we were not able to finish them at all of our study sites, and the results which we got in the end are a little bit messy. What is visible there is that birds and bats generally lead into the increase of arthropods when they are removed from the study system, but their effect is definitely not additive as was proposed in other studies. The numbers decreased similarly as both predators together, and at some elevations, birds were causing higher increase in abundance of arthropods, and sometimes bats did it. But we are still correlating the results with the biomass of the predators at the site. So in the end, it makes a sense, but like this, it looks a little bit messy. And when we look down to the herbivory, to the lower trophic levels, the results are not as I expected them. Based on the results from the review, I was expecting quite a big change in the herbivory after the removal of the ants. It was not happening. The only study site where it was happening was the study site at the lowest elevation, 200 meters above sea level. That was the only site where after the removal of the ants, the herbivorous damage increased nearly significantly. It was on the borderline. But what I saw is that the vertebrate predators removal is leading into the change of the herbivory. And in these main graphs, we were analyzing only the ficus species, which finished their distribution at 2,700 meters above sea level. But we were actually working even higher above the distribution of the ficuses. And if I extend the graphs like this to the higher two elevations, we could see that the effect of the vertebrate predators was detectable at the lowest elevations and at the three highest elevations, which would be corresponding with the middle hypothesis, generally that the cascades from the predators are making kind of a U-shape, being detectable in the most productive and the least productive study sites. Then why we were trying to explain why we didn't detect any effect of the ant removal on the herbivory, we returned back to the reviews, and we found out that all the exclosures where people tried to remove the ants were very short. Most of the experiments were shorter than 3 months, and these very short experiments resulted into the significant increase of the herbivory, while all the experiments which were a bit longer didn't result into any change. And because our experiments were 6 and 12 months long, then we simply think we observed the generally the dilution of the effect of the ant removal. And there might be another aspects also happening, but because in the data review we were not able to get into the data, so we will be looking into this in more details later on and investigating whether the spiders or other predators took some action here. The other aspects which are always important to consider when we are looking into the results of the exclosure experiments is the body size, because in all the exclosures we were conducting so far, we observed that in the resulting community on the excluded branches where the predators had no access, we observed that the mean arthropods are actually bigger, which is very simple. The predators are usually big, they are eating a big prey, and if they don't have the access, the bigger individuals are surviving happily. What was happening to us sometimes was that the big arthropods survived, but in the control branches, they were replaced by thousands and like many small individuals. So when we were looking into the abundances simply, we didn't see any change. But when we looked into the biomass and change in the biomass of the arthropods, then we saw quite a strong effect thanks to the change in the body size of the arthropods left on the branches. And this is of course very simple because, for example, at our lowest elevational study site, the bird had mean average body size 23 grams, and they were eating on the insect which was roughly 4 millimeter tall. And this is exactly the insect which were missing from the system from the control branches where the predators had access. The other aspects which might be affecting our results is also the composition and generally lifestyle of the arthropods which we are looking at the individual study site. For example, in Papua New Guinea, most of the caterpillars is rolling, and these rolling concealed caterpillars are not very often eaten by birds. So if there is a high proportion of the rolling concealed caterpillars in the study system and we exclude the birds, generally the effect of the bird exclusion is diluted and might not be detectable, while the ants feed on them happily. The other nasty things which were affecting our results were, of course, mesopredators. Along the gradient, the spiders represented about 20% of the diet of the birds, ants about 10%, and the herbivores represented around 60% of the bird diet. You can see along the elevational gradient that the ants were becoming less and less important in the diet, while the spiders were becoming more and more important. This is what we found after we were making the birds vomit and where we were identifying the insect in the diet samples which we obtained from them. And what we observed in the exclosure experiment pretty much matches these results from the diets. After we excluded the ants from the system, we have seen that the spiders took over the opportunity. They enjoyed the free space when the ants were missing, and they increased their abundances in contrast to control. And same, after the vertebrates were gone, they increased it a little bit again. So there was happening mesopredator release at some of the study sites, but not all of them. The other aspects are, of course, bottom-up control, which is significantly affecting all the aspects which we observed so far. And here I would like to show you the case study of our favorite Ficus haliana. Ficus haliana is very favorite because it grows along the nearly complete elevation of the gradient. It spans from the 200 to 2,700 meters above sea level, and its herbivory, standing herbivory on the control trees decreases with the increasing elevation. When we were looking into the constitutive defenses, we saw that they increase with the increasing elevation. And finally, when we return to the graph, which you have seen already once, we observed that the inducibility of the plant is higher in the lowlands. And when we spray the Ficus alliana with methyl jasmonate, it attracts more predators in lowlands than at the higher elevations. And here, when we assume that the abundances of the birds, as we have seen, do not change too much, and that the abundances of the insect do not change too much, we would expect that the difference between the control and the exclosure branches would be really stronger in the lowlands, as we have seen, than in the higher elevations when they generally overlap and didn't change significantly. This would imply that the Ficus alliana is protecting its own biomass a bit more by itself in the higher elevation, while relying on the protection by predators in the lower elevation. And we have generally two hypotheses. One of them is saying that the constitutive defenses should be stronger in the places where the enemies are abundant and should be decreasing towards the higher elevations where the insect is not so abundant. The other hypothesis is saying that the strong constitutive defenses should be in the places where the biomass is very expensive to do and where is it very expensive to replace. And seems that the Ficus alliana is the direct example of the second hypothesis, protecting itself strongly by the constitutive defenses at the higher elevation and having only the induced defenses in the lower elevation. And after we were looking into other plant species, other ficuses along the same elevational gradient, we found that all of them were actually following the second hypothesis, protecting themselves strongly at the harsh conditions, letting themselves to be protected by predators in the lowlands. But we have seen also in another experiment that the results might be affected by the access of the plants to nutrients. And when our student Honza Kolros was working on Tilia and Alnus in the temperate region, and again build the exclosures along them and fertilize them, we saw that in Tilia, which is very nasty, disgusting plant species by itself, the abundances of the insect increased after the plant was fertilized, which means that became more yummy, more insect arrived to it. While the alnus, which is a plant growing in very poor soils, not relying on not needing many nutrients altogether, the fertilizer itself didn't do anything to it. But when we excluded the predators from it, suddenly the abundances increased significantly, which means that the alnus is more dependent on the help of the predators, relying more on the induced defenses than the Tilia, which is limited by the nutrients. And because all these experiments were happening in the forest understories, somewhere dark below the canopies, and I was dealing with the predators, which I didn't see generally at all. And all the green foliage and all the predators were flying somewhere above my heads. I decided that the next research way to go is to look up to the forest canopies. The forest canopies are expected to host much higher diversity than the forest understories, and there should be also much stronger trophic cascades, according to the hypothesis, but it has never been tested before because the access to the canopies is very complicated. And because we are very lucky now, we have the chance to work along the potentially latitudinal gradient of the canopy cranes, which are standing in Tomakomai, in Japan, in China, in Papua New Guinea, in Kakoba, where the Kakoba crane is, and then we have other cranes in Tro, close to the cairns in Australia, and a Yukface facility close to the Sydney in Australia. There used to be also a crane in Lumber Hills in Malaysia when I was writing the grant proposal, but unfortunately the crane was shut down last year, so we didn't get to work there. But so far we have got 5 nice cranes where we could work with, get to the canopies and study how the predation changes there. And we conducted the experiment, which was very similar to the one which we were doing along the elevation gradient in Papua New Guinea. Again, we had the same 6 treatments, excluding ants, vertebrates, all predators, birds, and bats, but this time in the forest canopies and in the forest understories at the same time. Of course, we were not able to work on the fibrate ficuses anymore, but at each of the study sites, we pre-selected phylogenetically standardized species, which were also checked if they are abundant there enough on the plots, and we worked on them. And because the project is still underway and I can't really reveal too many results yet, I would like to summarize for now that we used lots of PVC pipes, which we shipped from Czech Republic to the 5 study sites. We used many football fields of the fishing netting, which we obtained in the countries, and built hundreds and hundreds of the small exclosures, which we applied on the branches in the canopy and on the saplings in the understory. So far, we collected about 84,000 spiders, 200,000 other insects, and we measured and remeasured 100,000 leaves at the beginning and at the end of the experiment. Similarly as in the previous study, we were also again surveying the predator communities, ants, birds, bats, by the song meters, by the capturing, by the mist netting. So we will have more or less complete information again what the predators eat and what is left after they can't eat from the branches. And here are some results which we obtained so far from the predation on the plasticine caterpillars. And to me, they were very surprising for the first sight. And I was wondering what is happening here, because against to our expectations, we observed the higher predation in the canopies only in the temperate region, here in Tomakomai in Japan and in the Yuk phase. At all tropical study sites in China, Kakoba in Papua New Guinea and Cairns, the predation on the plasticine caterpillars was lower in the canopies than in the understories. And this is exact opposite results which we would expect based on the expectations and hypotheses that the cascade should be stronger in the canopies. But when I was actually looking now into the bird data, it makes lots of sense, because the insectivorous birds are way more abundant in the temperate canopies, while they hunt mostly in the forest understories. And the majority of the birds living in the forest canopies in tropics are mostly frugivores. So the biomass of the insectivores is the richest in the forest understories. So it matches a little bit the biomass of the predators, but it's not expected results based on the hypothesis. So we will see how the results go like with the exclosures. And here are the results from the very first study site from Japan, which we finished as the first one. Here, when we look first at the canopy, control site, and exclosure, nothing happening according to our expectation. But when we excluded the vertebrates and all predators together, the abundances of the insect on the protected branches jumped up very significantly. I had to zoom into the results from the forest understories because they are a little bit lower and not so significant changes were observed there. But again, the pattern is quite clear. There is a control which is not significantly different from the effect of the exclosure of ants, but then all other predators led into the increase of the arthropods. So this is for now where I would like to leave you. I can't show more results now because we are keeping them for the publication. So please stay tuned and check as soon as we publish it. And because my time is also running out, I would like to make some conclusions. And generally, all my talk revealed that the interplay between the top-down and bottom-up control is really complex and works generally on the basis of characteristics of each plant species in the community. And some of these plant species can have different effects even on the level of the individuals, depending on how nutrient soil they are growing from. The cascades at different places depend also at the composition of the predators. Specifically, we found that it's very well correlated with the biomass. And there seems to be a very clear trade-off between the constitutive and induced defenses along the gradients. We found that vertebrates, especially birds, are very effective predators of arthropods. And again, it's corresponded well with the biomass of the insectivorous birds. Yet the cascades from the birds to plants were sometimes weak, thanks to the intraguild predation, and thanks to the birds eating both arthropods and predators in the same proportion. The trophic cascades from the insectivorous vertebrates along the Mount Wilhelm were detectable in the lowest and at the highest site of the Mount Wilhelm, which is in correspondence, which is against the theories that the cascade should be detectable only at the most productive, lowest elevational study sites. And finally, I would like to say that despite I work a lot with the plasticine caterpillars, I more and more believe that the method is not really suitable for estimation of the predation by ants. It more likely detects the abundance of the ants and their ability to try to bite into something rather than reflecting their ability to remove the real live insect from the study system. But the plasticine caterpillars correspond well with the predation of the birds along all the study sites which we were working at. And at this time, I would like to thank you very much for your attention, listening to my talk, and the big thanks belong also to my funders, to your European Research Council, to the grant agency of Czech Republic, and big thanks, like really big thanks goes to all the managers of the cranes where we were working last year, where we got stuck with the experiment. Michelle, Vinod, and Craig, who helped us a lot, saved our exclosures during the COVID times, and with the volunteers, they sent us all the samples back to Czech. And to all my assistants and students, PhD students from my lab, who work on the individual taxa and on the individual experiments. And thank you very much for your attention.
Okay, thank you very much, Katerina. Wojtek, would you please, coming back to here as society, Wojtek, would you please hold the session, the question of session, or I can restart with a question from someone who is on Facebook. So last question was from Katerina Campo. How do you manage a canopy exclosure to just exclude birds and not bats? Are they automated or is it done manually?
Okay, I have to admit that as you had seen in the end in the graphs, the exclosure was not possible in the canopies due to the safety regulations on the cranes, because it would require us to be on the crane at 5 a.m. and not a single manager would be doing that for us. I think the only study sites where we are considering that is in Papua New Guinea, where the regulations are not so strong, and there it will be done manually. And the thing is that the construction is from the PVC pipes, it's quite strong, and you can pull the netting along the construction. So we will have, unfortunately, here the results only from one study site, thanks to the rules.
Thank you. Then, Wojtek, can you hold the session so I can check on Facebook, please?
Yes. So we have Another question here about induced and constitutive defenses of plant species. Can they differ between populations of the same species according to ecological conditions, including response to herbivore pressure? From Eckhard Heyman.
Yes, I think they definitely can, because it's all regulated by the nutrient requirements of the plants. The constitutive defenses are generally very expensive, while the induced defenses are cheaper for the plant. So if the community is growing on the poor soils, it might be investing preferably into the induced defenses and can't afford the constitutive defenses as such. I hope I answer the questions correctly.
Okay, yeah, if there are any follow-up questions on your response, then please, anybody can write them again to Facebook or to the Zoom chat. In the meantime, I will move to the next question from Ute Radspiel. Can you explain a bit more what you mean by a dilution effect with regards to the differences in the effect of short-term versus long-term studies from the first part of your talk?
Okay, so this might be the competition between the spiders and ants, which is described from several study sites, many in Australia, but I believe there are other publications, which means that it takes some time until the spiders realize there is a free niche. So if you remove the ants immediately, you usually do it very effectively at the first few weeks. It takes some time to the spiders or other predators to realize there is a free space, and it works well. So this is the delusion. So over the longer time, the other predators might find a way out, or in actually. And the other aspect is that it's really difficult to exclude the ants effectively for a long time. So if you apply the tangle glue, it might work well for a few weeks, but then if you don't control correctly, the ants will get somehow in through the glue around. So this is what I meant by the delusion of the effect.
Okay, I see Ute nodding, so we can move to the next question. There are actually two very similar ones. Annemarie Wurz and Michael Stab, they both ask about what would be an adequate method to study predation by ants instead of dummy caterpillars. And again, second question is pretty much the same. What are alternatives to plasticine caterpillars?
No, like all the other methods, like any bait, generally the plasticine to me is a bait. It's a clay which consists of the fats and the salts, and these are things which the people studying ants are usually using as a bait. So exposing the plasticine, you are actually baiting the ants. So it really doesn't seem to express the predation as such. So I think that removal of the ants is really the way how to test what the ants are capable to remove from the system and what can escape and fight back.
Okay, I will just comment. I agree, but at the same time, there is a difference between attack rate, the predation pressure, and the response. The removal is a combined effect of what is the effect of the predators and what is the response of the populations of herbivores. So it's a slightly different parameter. I think we should still be trying to find some good bait which works with ants as well.
Yeah, I agree partly, but also I think the way how to go would be also maybe to install some cameras, camera traps if it's possible, because there is many ant species who don't respond to the plasticine caterpillars at all. Specifically, Ecophylla. Ecophylla is walking around the plasticine like around. Despite the Ecophylla represents the biggest, the most abundant species in cairns, it doesn't touch the caterpillars at all. And it's happily sitting on it and defending that. So we would need to know more about the behavior of the individual and species to the bait as such.
Mm-hmm. Okay, I have one more question here in the chat. Why is the effect of ant removal lost over time? Is it replacement by other predators only, or there is something else? That's one question. And then second is, what is going on with the arthropod abundance at 700 meters? So I think that refers to what you briefly mentioned about the middle elevation effect.
Yes, actually, yeah, I think I responded to the first part of the part about the ants dilution effect, and what's going on with the arthropods at 700 meters. I totally don't know. Like, maybe it's a question more up to you. My sample size is really smaller than yours, like to Wojciech's. It might be simply that we didn't survey large enough hectares of the forest, might be some slight increase, but what we observed on the saplings, there really was slightly more arthropods and also higher herbivory at 700 and 1200 meters above sea level. So the overall herbivory was making a hump shape a little bit, and it correlated with the abundance of the arthropods there on the saplings which we surveyed.
Okay, I will return again to the very popular topic of baits for ants, because there is a follow-up question from Nina Farvik. So would you install the living prey in front of cameras, or how should the camera measurement work?
Yes, exactly. That's what I would do. Unfortunately, the camera traps do not work for ants, so it would have to be the cameras, CCV cameras reporting, measuring the movements of the ants around a real bait. And if it's a real bait, you could then see when it's escaping.
Yes, I think I agree. This would be logistically very difficult again, because if the recording is continuous, you have to recharge cameras. We had a project doing exactly that for bird nest predation in Papua New Guinea, and that worked well, but was a constant logistical demand on the experiments. So this would be the same. Yeah, there is a mention, there is a comment that Olivier Hardy's group is developing cameras for invertebrate pollinators. So that's probably, yeah, there are some other groups doing the same. So yes, I think that's probably the way to go. Okay, I think we have 1 minute left, which is fine, because we have just finished all the questions. I'm just checking the Facebook. But there is nothing new as well. So I think we can thank everybody for discussion, and unless you want to have some, maybe you can have some closing remarks, and then we can close the plenary.